In 2012, a quiet genetic study did something no army, no empire, no revolution had ever managed to do. It cracked open the sealed narrative of human history. Scientists analyzing Ethiopian DNA weren't searching for headlines. They were mapping ancestry, tracing migrations, filling in gaps. But what they uncovered wasn't a gap. It was a fracture line running through everything we thought we understood about where we come from. Inside Ethiopian genomes, researchers found signals that should not have existed. Genetic segments that did not match the established branches of modern humans. Not Neanderthal, not Denisovan, not any known archaic group. These sequences hinted at encounters with populations separated by unimaginable distances and separated by time itself. While much of Africa was assumed to have evolved in relative continuity, Ethiopia appeared different. It looked less like a simple origin point and more like a vault, a time capsule. But the true shock wasn't the presence of strange DNA, it was the pattern. Ethiopian DNA carried layer upon layer of ancient interaction, as if humanity had returned to this land again and again, leaving genetic signatures each time. The Horn of Africa was not just a starting line, it was a crossroads, a magnet, a convergence zone across millennia. For decades, the dominant model was clean and linear. Humans evolved in Africa. A small group left roughly 60,000 to 70,000 years ago. They spread across Eurasia, meeting Neanderthals in Europe and Denisovans in Asia. That was the story. Elegant, ordered, finished. Ethiopian DNA complicated that story, because while Europeans carry about 2% Neanderthal DNA and some Asian populations carry Denisovan ancestry, Ethiopians preserve something far more elusive: genetic traces of what scientists now call ghost populations, archaic humans who left no confirmed fossils, no tools we can definitively assign, no skeletal remains labeled in museum drawers. They exist only as patterns in living cells. These ghost signatures suggest that Ethiopia was not merely the cradle of humanity, it was a meeting ground for multiple human lineages. Populations separated for hundreds of thousands of years may have converged there, interbreeding and blending before dispersing again. This challenges more than migration timelines. It challenges the simplicity of our origins. It suggests that human history was not a straight line radiating outward from Africa. It was a web, a series of departures and returns, and Ethiopia appears at the center of that web. What you are about to see is not speculation layered over myth. It is genetics layered over time, evidence that forces archaeologists, historians, and evolutionary biologists to reconsider assumptions once treated as fact. Because Ethiopian DNA does not just tell the story of Africa, it tells the story of repeated contact, of forgotten branches of humanity, of genetic memories that survived where bones did not. And if the evidence holds, then the most forbidden chapter of human history was never erased. It was encoded in Ethiopia.
Neanderthals, Denisovans, and Ethiopian ghost populations. Every human outside Africa carries a genetic souvenir from the distant past. Around 2% of European and Asian DNA comes from Neanderthals. In parts of Asia and Oceania, Denisovan ancestry lingers in measurable amounts. These fragments are the molecular proof of encounters that happened roughly 50,000 to 60,000 years ago, when modern humans expanding out of Africa met other human species already living across Eurasia. For years, that was the framework. Modern humans left Africa, they met archaic humans elsewhere, they interbred. End of story. But Ethiopian DNA does not fit neatly inside that model. When researchers began sequencing Ethiopian genomes at higher resolution, they noticed something unusual. The archaic segments embedded in Ethiopian DNA did not align cleanly with Neanderthals or Denisovans' reference sequences. Instead, certain fragments appeared older, more divergent, as if they belonged to a lineage that separated from the ancestors of modern humans hundreds of thousands of years before Neanderthals and Denisovans split off. In other words, Ethiopians Ethiopians seemed to carry genetic echoes of a population that science has never formally identified. These so-called ghost populations are inferred through statistical modeling. Scientists compare DNA across global populations and look for segments that cannot be explained by known lineages. When the numbers refuse to balance, when inheritance patterns do not match expected evolutionary trees, the only explanation is admixture with a group we have not yet discovered in the fossil record. Ethiopian genomes consistently show evidence of such admixture. Some models estimate that between 4% and 7% of Ethiopian ancestry may derive from an archaic African lineage that diverged from modern humans roughly 400,000 to 500,000 years ago. That divergence predates the commonly accepted emergence of Homo sapiens as a species. It implies that as anatomically modern humans evolved, they did not do so in isolation. They were interacting with other human groups still living within Africa. This reshapes a central assumption. For decades, archaic admixture was framed primarily as an out-of-Africa phenomenon. Modern humans left, met Neanderthals and Denisovans, and absorbed small genetic contributions. But Ethiopian DNA suggests something equally complex was happening inside Africa itself. The African continent was not a single uniform population slowly transforming into Homo sapiens. It was likely home to multiple semi-isolated human groups evolving in parallel. Some of these groups eventually vanished, leaving no skeletal remains confidently attributed to them. Yet their genetic fingerprints persist in Ethiopia. Why Ethiopia? Geography may offer part of the answer. The Ethiopian highlands sit at a crossroads between sub-Saharan Africa, North Africa, and the Arabian Peninsula. Climatic fluctuations over hundreds of thousands of years would have turned this region alternately into a refuge and a corridor. During arid periods, highland environments could sustain populations when surrounding areas became inhospitable. During wetter phases, migration routes would reopen, allowing separated groups to reconnect. But geography alone does not explain the depth of divergence observed in Ethiopian ghost ancestry. The statistical models imply long periods of separation followed by recontact. That means these populations were not simply neighboring tribes, they were lineages evolving apart for immense spans of time before merging again. This pattern resembles what happened between modern humans and Neanderthals in Eurasia. Yet in Ethiopia, the interaction appears older and potentially more complex. Instead of a single pulse of admixture, some studies suggest multiple waves. Even more intriguing is the distribution of this ancestry. It is not confined to a single Ethiopian ethnic group. Variations of archaic African admixture appear across diverse populations within Ethiopia, though at differing proportions. That suggests these events occurred deep in the past, before the formation of many contemporary cultural identities. The implications are profound. If modern humans interbred with archaic African populations long before leaving the continent, then Africa was not merely the birthplace of Homo sapiens. It was a mosaic of human forms, exchanging genes over hundreds of thousands of years. The boundaries between species may have been more porous than we imagined. Ethiopia, in this framework, becomes a repository of that complexity, a place where ancient lineages overlapped and where genetic diversity accumulated rather than vanished. And this is only the beginning, because when we move from abstract ghost populations to specific archaeological discoveries in Ethiopia, the timeline grows even more unsettling.
The cradle of humanity. Long before genetic sequencing, Ethiopia had already earned its place in human history. In 1974, in the Afar Depression, paleoanthropologists uncovered the fossilized remains of a 3.2 million-year-old hominin later named Lucy. She was not human in the modern sense, but she stood upright, she walked, she belonged to a lineage that would eventually lead to us. Her discovery cemented Ethiopia as one of the earliest chapters in the human story. But Lucy was only the prologue. In 1967, fossils from Omo Kibish in southern Ethiopia were dated to roughly 195,000 years old. Later discoveries at Herto pushed anatomically modern human remains in Ethiopia back to around 160,000 years ago. These dates placed Ethiopia at the center of Homo sapiens emergence. For decades, the conclusion seemed straightforward. Modern humans evolved here, then dispersed. Genetics complicated that simplicity. When scientists began analyzing the DNA of contemporary Ethiopian populations, particularly groups like the Afar, who still inhabit regions near Lucy's discovery site, they found extraordinary diversity. Certain Y chromosome lineages in Ethiopia belong to some of the oldest branches known in living humans. These lineages diverged very early in our species' genetic tree, preserving signals close to humanity's common paternal roots. This does not mean Ethiopians are more ancient than others in a hierarchical sense. All modern humans share common ancestry, but it does mean that some Ethiopian populations retain lineages that split off near the base of our shared genetic history, lineages that disappeared elsewhere through bottlenecks and migrations. Even more striking is the overall level of genetic diversity found within Ethiopia. Studies have shown that a single Ethiopian region can contain more genetic variation than entire continents outside Africa. This is consistent with a broader pattern. Africa holds the highest human genetic diversity globally, but Ethiopia stands out even within Africa. Why would such diversity persist there? One explanation is continuity. Populations in Ethiopia may have experienced fewer complete replacements than regions affected by later mass migrations. Instead of being overwritten, older lineages blended with newcomers. Each wave added new layers without erasing the old. Then there is the evidence for archaic African admixture. Computational analyses comparing Ethiopian genomes to other African populations suggest gene flow from deeply divergent lineages. These lineages separated from modern human ancestors hundreds of thousands of years ago. Their DNA signatures appear embedded within Ethiopian populations in measurable proportions. Unlike Neanderthals, these archaic Africans left no confirmed fossil identity. They are detected only through statistical modeling, yet the signal is strong enough that independent studies have converged on similar conclusions. Somewhere in Africa, ancient human groups persisted alongside early Homo sapiens and interbred with them. The Ethiopian highlands, with their varied ecology and relative climatic stability, could have served as long-term refuges. During periods of severe drought across Africa, high-altitude regions with consistent rainfall would have remained habitable. Populations isolated elsewhere might have converged there during favorable periods, creating cycles of separation and reunion. This possibility reshapes the out-of-Africa model. Rather than a single cohesive population evolving uniformly and then dispersing, Homo sapiens may have emerged from a network of interconnected African populations exchanging genes intermittently. Ethiopia, positioned between Eastern Africa and the Arabian Peninsula, would have been central in that network. There is also growing evidence that migrations were not strictly one way. Archaeological finds in the Levant suggest modern humans left Africa as early as 100,000 years ago in earlier waves that may not have led directly to all non-African populations today. Climate shifts could have forced some groups back into Africa, potentially returning through regions near the Horn. If such return migrations occurred, Ethiopia would again have been a natural gateway. Over tens of thousands of years, departures and returns could have layered additional complexity onto local genomes. All of this reinforces a single idea. Ethiopia is not just a birthplace frozen in prehistory. It is a dynamic intersection, a place where human lineages repeatedly met, separated, and rejoined.
The genetic enigma. Around 3,000 years ago, something extraordinary happened in Ethiopia. Genetic data across multiple Ethiopian populations points to a massive admixture event. This was not a slow blending over many centuries. The signal appears sharp, sudden, and widespread. A large influx of Eurasian-related ancestry entered the region and spread rapidly. What makes this unusual is its scale. In several Ethiopian groups, close to half of their ancestry traces to this ancient Eurasian source. Yet this ancestry does not resemble recent colonial or medieval mixing. Genetic modeling dates it to roughly the first millennium BCE. This timeline overlaps with the rise of early complex societies in the Horn of Africa and increasing connections across the Red Sea. Archaeological evidence shows contact between southern Arabia and northern Ethiopia during this period. Writing systems, architectural styles, and trade goods suggest sustained interaction. Genetics confirms that interaction was not superficial. When scientists compared Ethiopian genomes to populations in the Near East, they found affinities not to modern Arabian groups alone, but to ancient Near Eastern populations. The genetic components appear closer to groups that lived thousands of years ago than to many present-day populations. This implies that the admixture event reflects ancient migrations rather than recent trade alone. One of the clearest examples appears among Ethiopian Semitic-speaking populations. Their languages belong to the Semitic branch, related to Hebrew and Arabic, yet they evolved independently within Africa. Linguistic analysis suggests an early introduction followed by long local development. Genetic studies align with this pattern. The same populations that speak Ethiopian Semitic languages often show significant Eurasian ancestry dating back around 3,000 years. Genes and language appear to move together. This does not indicate simple replacement. The Eurasian-related ancestry did not erase earlier African lineages. Instead, it merged with deeply rooted African genetic backgrounds that had already accumulated archaic components. The result was not dilution, but layering. Ethiopia became a place where some of the oldest African lineages combined with ancient Near Eastern ancestry in a single population. High-altitude adaptation provides another clue to Ethiopia's uniqueness. The Ethiopian highlands rise above 2,500 meters in many areas. Living at these elevations presents physiological challenges due to reduced oxygen levels. Different human populations have solved this problem in different ways. Tibetan highlanders possess genetic adaptations affecting hemoglobin regulation. Andean populations exhibit increased red blood cell production. Ethiopian highlanders, however, show a distinct pattern. Certain variants in genes associated with oxygen transport and cardiovascular efficiency appear unique to East African highland groups. These adaptations likely developed over thousands of years of continuous residence at altitude. When Eurasian ancestry entered the region, it encountered populations already finely tuned to highland environments. The resulting gene pool combined local physiological adaptations with incoming genetic diversity. Ethiopia's medical genetics also reveals unusual combinations. Variants associated with resistance to certain infectious diseases common in Africa appear alongside variants linked to conditions more typical in Eurasian populations. In some cases, protective mutations coexist in ways that reduce negative side effects seen elsewhere. For example, specific gene variants that offer protection against parasitic diseases are present in Ethiopia, but their expression patterns differ from other African regions. Researchers are still investigating how combinations of mutations may interact to modify disease risk. Religious and cultural history adds further layers. Ethiopia adopted Christianity in the 4th century CE, making it one of the earliest Christian states. At the same time, Islamic communities developed in eastern regions through trade connections. Jewish communities, known as Beta Israel, maintained distinct religious traditions for centuries. Genetic testing of Ethiopian Jewish populations revealed predominantly Ethiopian ancestry with detectable links to ancient Middle Eastern lineages. These markers are not identical to modern Middle Eastern populations, but align with older genetic patterns reconstructed from ancient DNA studies. The result is a complex picture. Ethiopian populations are neither isolated relics nor simple products of recent mixture. They represent a convergence of ancient African diversity, archaic African contributions, and substantial early Eurasian admixture. The admixture event around 3,000 years ago affected multiple regions within Ethiopia. Its genetic signature appears across Christian, Muslim, and Jewish communities alike. This suggests a widespread demographic transformation rather than isolated intermarriage. Yet even this dramatic shift
did not erase earlier history. Beneath the Eurasian layer remains a foundation of some of the deepest human lineages on Earth. Ethiopia's genetic profile is not defined by a single origin. It is defined by accumulation, and the next layer of that accumulation emerges through the rise of powerful civilizations that connected Ethiopia to 3 continents at once.
Lost civilizations long before modern borders divided continents, the Horn of Africa stood at the center of global exchange. The Aksumite Empire, rising around the 1st century CE, became one of the great powers of the ancient world. It minted its own coins, built towering obelisks, and controlled trade routes stretching from the Mediterranean to India. Trade was not only about gold, ivory, and incense, it was also about people. Genetic studies show that Ethiopian populations contain markers linking them to regions across the Red Sea, the Levant, parts of southern Europe, and even South Asia. These signals are not uniform. They appear in layers, consistent with repeated contact over centuries rather than a single event. The Red Sea functioned less as a barrier and more as a bridge. Maritime trade connected Aksum with Arabian kingdoms. Archaeological evidence documents South Arabian inscriptions in northern Ethiopia and architectural parallels between regions. Genetic data supports sustained interaction during this period, reinforcing earlier admixture events and adding new strands. Further north, along the Nile corridor, connections extended toward Nubia and Egypt. Ancient trade and political alliances linked these regions for centuries. Modern genetic comparisons reveal affinities between certain Ethiopian and Nubian populations, suggesting shared ancestry shaped by long-term contact. These relationships were not static. As empires rose and fell, movement continued. Merchants, soldiers, scholars, and religious figures traveled across these networks. Each wave left subtle genetic traces, contributing to Ethiopia's layered ancestry. The medieval period added new dimensions. Islamic expansion reshaped trade across the Indian Ocean. Ports along the Horn became hubs in a vast commercial system linking East Africa, Arabia, Persia, and India. Genetic signals associated with these regions appear in varying degrees among Eastern Ethiopian populations. In some areas, the genetic imprint aligns with historical accounts of merchant settlements and intermarriage. These contributions are measurable, yet blended into the broader Ethiopian genetic framework, rather than forming isolated enclaves. The Harla civilization, once prominent in eastern Ethiopia, left behind stone cities and trade centers before declining in the 16th century. While their material culture faded, genetic continuity suggests that their descendants merged into surrounding populations rather than disappearing entirely. The pattern repeats across regions. Cultural change did not always mean population replacement. More often, it meant absorption. In the highlands, monumental achievements such as the rock-hewn churches of Lalibela in the 12th and 13th centuries reflected connections with the wider Christian world. Pilgrims, artisans, and clergy traveled between Ethiopia and other Christian centers. Though smaller in scale than earlier admixture events, these contacts added further genetic diversity. What distinguishes Ethiopia is not merely the presence of foreign genetic components. Many regions of the world experience trade and migration. The difference lies in continuity. Instead of cycles of complete replacement, Ethiopia often shows evidence of integration. Incoming groups did not eliminate prior populations, they merged with them. Earlier lineages persisted beneath new layers. Over time, this created complex genetic mosaics within the same communities. This accumulation is visible in disease resistance patterns. Protective variants against malaria, common in sub-Saharan Africa, coexist with genetic traits associated with populations from the Near East and beyond. The combination reflects centuries of exposure to diverse environments and pathogens. Such layered diversity may have provided resilience. Historical records describe epidemics and invasions that affected neighboring regions severely. Ethiopia endured many of these pressures while maintaining demographic continuity. Geography played a role. Mountainous terrain offered natural defense, but genetic resilience, built through accumulated diversity, may have contributed as well.
well. Ethiopia's position between Africa, the Middle East, and the broader Indian Ocean world ensured continuous interaction. Yet, unlike many trade hubs that experienced repeated population turnovers, Ethiopia's core populations show deep continuity stretching back thousands of years. The result is a genetic archive shaped by empire, religion, commerce, and migration. And as modern science advances, this archive is revealing patterns that extend beyond the past and into questions about human biology and potential.
The future in the past. When the COVID-19 pandemic spread across the globe in 2020, scientists began analyzing why outcomes differed between populations. Many factors influenced severity: age distribution, public health systems, reporting accuracy, and pre-existing conditions. But researchers also examined genetic variation, especially genes related to immune response and cellular receptors used by the virus. Within this broader research, African genomic diversity drew increasing attention. Ethiopia, already recognized for unusually high genetic variation, became part of that discussion. Studies exploring variants in immune-related genes, including those affecting inflammatory response and viral entry pathways, highlighted differences in frequency between Ethiopian populations and other global groups. These variants were not new mutations. Many were ancient, embedded within lineages that had persisted for thousands of years. Some appear connected to long-standing exposure to diverse pathogens. Over time, repeated disease pressures may have shaped combinations of immune-related genes in ways that differ from other regions. This does not mean immunity to specific viruses is predetermined. Disease outcomes depend on many interacting variables, but Ethiopian genomes demonstrate how long-term genetic diversity can influence biological response patterns. The broader significance extends beyond one pandemic. For decades, the human reference genome used in research was based primarily on individuals of European ancestry. As more African genomes were sequenced, scientists discovered that significant portions of global genetic diversity were missing from standard references. Ethiopian populations contributed numerous previously undocumented variants. Some of these variants occur in regions associated with metabolism, cardiovascular function, and neurological development. Others affect how genes are regulated, rather than altering protein structure directly. In certain cases, sequences identified in Ethiopian genomes represent insertions or structural differences not well captured in earlier models. This expanded understanding has practical implications. Medical treatments and risk predictions based solely on limited reference data can overlook variation present in other populations. Including Ethiopian genetic data improves the accuracy of global biomedical research. Athletic performance offers another example of complex genetic interaction. Ethiopian long-distance runners have dominated international competitions for decades. High-altitude upbringing contributes to endurance capacity, but genetic research indicates that no single performance gene explains success. Instead, multiple small-effect variants influence oxygen utilization, muscle fiber distribution, and energy metabolism. The advantage appears combinatorial. It reflects how numerous subtle genetic factors shaped by long-term adaptation and admixture can interact. This pattern mirrors Ethiopia's broader genetic structure. Rather than extreme specialization in one trait, Ethiopian populations often show layered combinations of adaptations. Recent large-scale sequencing initiatives within Africa aim to document this diversity systematically. Ethiopian participation in such projects expands knowledge not only of local populations, but of humanity as a whole. Because many of the deepest branches of the human genetic tree remain rooted in Africa, studying Ethiopian genomes helps reconstruct early human evolution more accurately. There are also ethical dimensions. As genomic research expands, questions of data ownership and genetic privacy become increasingly important. Nations with high genetic diversity represent valuable scientific resources, ensuring that research benefits local communities while protecting individual rights is essential. Ethiopia has taken steps to regulate genetic research and preserve national oversight.
Balancing open scientific collaboration with sovereignty over genetic data remains a global challenge. Beyond medicine and policy, Ethiopian DNA raises broader questions about human potential. If ancient admixture events combined deeply divergent lineages and later migrations layered additional diversity, then Ethiopian genomes represent one of the most complex mosaics on Earth. This mosaic does not signify fragmentation, it signifies accumulation. Over hundreds of thousands of years, populations met, separated, and met again. Adaptations developed in one context blended with those from another. Traits shaped by high-altitude survival merged with those influenced by arid environments, tropical pathogens, and temperate zone exposures. The result is not a single narrative, but a composite. Ethiopia's genetic history demonstrates that human evolution did not proceed through isolation alone. Interaction mattered, diversity compounded, and in at least one region of the world, that compounding remained visible rather than erased. The story encoded in Ethiopian DNA is not a closed chapter, it is ongoing. Each generation carries forward combinations forged in ancient encounters. Each new scientific discovery reveals patterns once invisible. The forbidden chapter of human history was never destroyed, it was written in code, and that code is still alive.